and welcome to Common Hour. Today's Common Hour speaker, Dr. George Koob, is an internationally recognized expert on alcohol and stress and the neurobiology of alcohol and drug addiction. Few have really dedicated the entirety of their career to studying a single important problem in public health and neuroscience with the vigor that Dr. Koob has. His research is focused on understanding the neurochemistry and the neurobiology of emotional behavior and how maladaptation in the neural circuitry that underlies our emotional behavior can give rise to alterations in motivation that lead to addiction. Dr. Koob is currently the director of the National Institute on Alcohol Abuse and Alcoholism of the National Institutes of Health. As the NIAAA director, Dr. Koob oversees a wide range of alcohol-related research, including neuroscience, genetics, epidemiology, prevention, and treatment. After completing his undergraduate degree in zoology, Dr. Koob received his PhD in behavioral physiology from the Johns Hopkins University. He spent most of his career at the Scripps Research Institute, which is known internationally as a center of excellence in neuroscience research, especially with regard to addiction. At Scripps, Dr. Koob served as co-founder and director of the Pearson Center for Alcoholism and Addiction Research. He's also professor and chair of the Scripps Committee on Neurobiology of Addictive Disorders. He's also been the recipient of numerous teaching awards at the University of California, San Diego. Dr. Koob began his career studying the neurobiology of emotion, including how the brain processes reward and stress. His contributions have advanced our understanding of the anatomical connections of the emotional systems and the neurochemistry of emotional function. This background led to investigations into why certain alcohol drinkers, but not others, transition to addiction, and how the brain and the body respond to alcohol consumption. Dr. Koob's work has significantly broadened our understanding of the neural circuitry associated with the acute reinforcing effects of alcohol and other drugs of abuse, and the adaptations of the reward and our stress circuitry that leads to addiction. He is the author of more than 600 peer-reviewed scientific journal articles and co-author of The Neurobiology of Addiction, a comprehensive review of over 50 years of neurobiological research on addiction. Dr. Koob is the recipient of many prestigious awards, including the Daniel Efron Award for Excellence in Research and the Axelrod Mentorship Award from the American College of Neuropsychopharmacology, in addition to numerous other awards. Dr. Koob's lecture today is sponsored by our HHMI-funded Leadership in Biomedical Research and Medicine seminar series, and we're proud to have him join us today. Before we lend him a warm welcome, I'd like to thank Carla Brigatti for her keen coordination of this event and others in our Leadership in Biomedical Research and Medicine seminar series. So please join me in a warm F&M welcome for Dr. George Koob.
Well, thank you very much, Robert and Carla, for inviting me here. It's a pleasure to be at Franklin and Marshall. As many of you know, my son is here and making all the noise over here in the left-hand corner. So I guess I should start by telling a story about my son. So you may wonder, what is a California kid doing in a place like this? And there are two pieces to the story. One is one of the professors here for many years was Charles Heiser, who is one of my former postdocs. And so Charles actually is the one who enticed Cameron with all the wonders and wonderfulness of Franklin and Marshall. But earlier than that, teenagers always like to act out in some way. And one of Cameron's great acting out manifestations that I never understood, but now makes a little more sense, is that he would go to high school at a surf surfing high school 3 blocks from the ocean in Southern California wearing a 3-piece suit every day. Okay, I have lots of other Cameron stories, but I'm not going to tell them. One of the things I want to impress upon you is the cost and scope of addiction and how serious it is as a problem. And this slide shows you that the prevalence is millions of Americans are afflicted with this disease. This incorporates drugs, illegal drugs as well as alcohol. And the cost to society is about $500 billion a year. And alcohol is, of course, one of the major contributors to that cost. And what I like to tell lay audiences is if you go to, certainly in San Diego County, and I assume it's the same in Lancaster County, if you go to the emergency room on a Friday or Saturday night, you are going to find that 50% of the people there are there because of a drug or alcohol problem. And the other thing I'll impart upon you as young people is that if you treated alcohol and drug addiction, you would save untold quadrillions of money in healthcare costs. So think about that when you start having to pay the bills yourself and you get a job. And one of the things we know in the alcohol field is that probably less than 20% of anyone with an alcohol use disorder gets any treatment whatsoever. So it is one of the challenges I face as director. Now, the title of my lecture today is actually, how do I see insights into these disorders, and what does it tell us about how the brain works, and life, liberty, and the pursuit of happiness? And that's where we're going to go today with this lecture. So I started off my career, the first 15 years of my career, trying to figure out how we felt good. And I like to say as I got older, I've been trying to figure out how we feel bad. And today's lecture really focuses on those two issues. So all of you have probably some idea of what reward is. I defined it down here as a stimulus, and in the case of drugs, it would be a drug that increases the probability of a response. It usually has a positive hedonic connotation. In other words, it makes you feel good. And most of you probably have some idea of what a stress is, and I particularly like this definition from Susan Birchfield: anything that causes alteration of psychological homeostatic processes. And in fact, physical stressors can activate your body's stress response, but many of you know that psychological stressors are much more effective, and sometimes physical stressors don't elicit those responses unless there's a psychological component to it. In other words, fear or anxiety. And so what does this have to do with the Roman god Janus? Well, he was the god of doors, of passages, transitions. His two faces look to the future and look to the past. He would be the god of transitions from young to old, from living to death. He was a statue was put at the doors of the gates of Rome to prevent invasion. And so reward and stress represent different components of the transitions in our brain that lead and perpetuate addiction. And I'm going to make the argument that they're intimately related, and that's something I think that's not commonly understood. And when I talk about stress in this context, I'm talking about negative emotional states in general, one of which is pain, negative emotional pain, as well as physical pain, but also emotional pain. I think that's important. So first of all, we're going to have a very brief lesson in neurobiology and neuropharmacology. So I want to tell you about the reward systems in the brain. I don't know whether you know it or not, but you have a reward system. So does a lizard, okay? And so when that lizard hits the fly with its tongue and ingests it, presumably his reward system is being activated. This is actually the reward system in the brain of a rodent, all right? This is a rat brain. And if you're anatomically challenged, the way you know it's a rat brain is there's no cortex, right? And that's a good thing. Do you really want rats running around with a cortex the size of our cortex? But the critical structure is a structure, it's an old structure phylogenetically, and even lizards, as I said, have it, and it's called the medial forebrain bundle. And in a galaxy far, far away, in a time far, far away, in 1954, two researchers in Canada, one an American, one a Canadian, were put electrodes in the brain of rodents and discovered that when they stimulated the brain of the rodent kept going back to the same place. And this is actually a brain from my PhD thesis at Hopkins. And the way I got this brain was that I actually anesthetized a rat, walked across the street to the radiology department at Hopkins in the medical school, and I said, hey guys, will you x-ray my rat? And they said, sure, and they ran it through the x-ray machine. If I did that today, you can only imagine what would happen. But the electrode is in the brain, deep in the structure of the medial forebrain bundle, and rats will press levers 100 times a minute. They'll jump over barriers. They'll go without food for quite a long time just to get this stimulation, and they have huge smiles on their faces, all right? So what causes this reward? How does this happen? How do these neurons work? What's in the medial forebrain bundle that makes you feel good? And this is a human brain now, and you can see that the medial forebrain bundle is in the same location, down in what I call the reptile brain, and of course, the human brain has this huge cortical mantle. And thank your lucky stars for that, because that's what separates us from rodents to a large extent. Now here's the basic lesson in neuropharmacology. I know most of you have had biology at some point, you probably had it in high school, but I just want to remind you that we have billions of neurons in our brain. They are the method of communication, and they go from one structure to another, and they're wired up in a kind of wiring diagram. And I use this as just a quick example to remind you that these neurons use transmitters to communicate. So electricity moves down the neuron, and you can think of it almost like a wire, a neuron, but ultimately it comes to a point where that electricity triggers the release of a chemical. And that chemical is shown here. We're using dopamine as an example, which is one of the reward transmitters. And you can see it's crossing the synapse and triggering another electrical change in the neuron down the line. And you've now had a course in neuropharmacology, all right? It's really that simple. There are millions, billions of these neurons in our brain. They're color-coded in the sense that they all have different neurotransmitters and they do different things. And you have to understand that basic precept to understand the rest of my lecture, that the brain is a wiring diagram, that it has specific chemicals in it, and they, in some cases, do specific things. So let's quickly talk about the reward system. So what I've argued is that the dopamine system, illustrated here, is at the beginning of that medial forebrain bundle. It starts back here in the hypothalamus, posterior to the hypothalamus, and these neurons project forward to an area of the brain that we call the basal ganglia, which I'll talk about in a minute. So when you find something good in the environment, there is a big phasic release of dopamine. It could be a member of the opposite sex. It could be, depending on your orientation, a member of the same sex. It could be the pizza over there when you're hungry. Any of these things are going to trigger initially a release of dopamine, and they're going to link up to the common hour, and you're going to remember next time you come in here that there was pizza for the common hour, and you're going to get another surge of dopamine release, which is going to keep you coming back to common hour. All right? But there's another transmitter that's also intimately involved in making us feel good. There's more in the realm of satiation. So when that pizza hits your stomach and the glucose and that good taste goes up into your brain, there's probably a release of endorphins or opioid peptides. And they're the little blue guys here that also interface with the medial forebrain bundle. In other words, they all interact together. That's your reward system, and we learned about that through our study of drugs of abuse. And we all have a reward system, and presumably we all have a reward system functioning at all times to make us feel good, tiptoe through the tulips, and have a happy life. But we also have a brain stress system. And so I want to tell you about the other side, or what I call the dark side of emotionality or addiction. And the best way to tell you about the brain stress system Well, first, I defined it earlier as any nonspecific demand on the body, the psychological demand, but the old definition was stress is any nonspecific result of any demand upon the body in any form. And it was measured in the old days as a release of glucocorticoids from your adrenal gland. And glucocorticoids we'll talk about in a minute, but glucocorticoids are critically important for glucose metabolism, for suppressing your immune system, for increasing blood glucose and getting it to your brain, all the things you need to mobilize fight or flight when a stressor comes toward you. But nobody described the neurochemistry and neurobiology of stress better than Kurt Vonnegut in The Breakfast of Champions. So I want you to read this with me. And I want you to know that this was written in 1973, almost 10 years before what he wrote here actually was discovered at the Salk Institute. So it goes like this. I saw Kazak out of the corner of my right eye. His eyes were pinwheels, his teeth were white daggers, his slobber was cyanide, his blood was nitroglycerin. He was floating toward me like a zeppelin hanging lazily in the air. My eyes told my mind about him. My mind sent a message to my hypothalamus, told it to release the hormone CRF into the short vessels connecting my hypothalamus and my pituitary gland. The CRF inspired my pituitary gland to dump the hormone ACTH into my bloodstream. My pituitary had been making and storing ACTH for just such an occasion. And nearer the zeppelin came, and some of the ACTH in my bloodstream reached the outer shell of my adrenal gland, which had been making and storing glucocorticoids for emergencies. My adrenal gland added the glucocorticoids to my bloodstream. They went all over my body, changing glycogen into glucose. Glucose was muscle food. It would help me fight like a wildcat or run like a deer. And nearer the zeppelin came. My adrenal gland gave me a shot of adrenaline, too. I turned purple as my blood pressure skyrocketed. The adrenaline made my heart go like a burglar alarm. It also stood my hair on end. It also caused coagulants to pour into my bloodstream, so in case I was wounded, my vital juices wouldn't drain away. Everything my body had done so far fell within the normal operating procedures for a human machine. But my body took one defensive measure, which I am told was without precedent in medical history. It may have happened because some wire short-circuited or some gasket blew. At any rate, I also retracted my testicles into my abdominal cavity, pulled them into my fuselage like the landing gear of an airplane, and now they tell me that only surgery will bring them down again. Believe it or not, male adolescent rats will retract their testicles into their abdominal cavity, but it doesn't take surgery to bring them down again. It just takes a cute female rat to show up. What this led to, and it's a momentous part of my career because I was at the Salk Institute when this discovery was made, but Wiley Vale and his collaborators at the Salk Institute, Catherine Riviere, Jean Riviere, Joachim Spies, discovered this peptide and characterized this peptide long before cloning, I might add. It was in 1981. And what we subsequently discovered is that Vonnegut had it right. It's an extraordinary story. I, to this day, don't really know how he knew that, except that he had family members who were in the medical profession. But in any event, what Vonnegut argued, and what I just told you from his quote, is that CRF is our stress neurotransmitter. This peptide in your hypothalamus and outside of your hypothalamus in an area of the brain called the amygdala drives your stress response, the whole stress response, all of it. And it's described on this slide. There are three components. There's the pituitary adrenal response, which is the glucocorticoids, which you heard what Vonnegut said about that. But there's also a brainstem sympathetic response, which is increases your blood pressure, gets your heart rate really banging away. And then there's an emotional response. So if you're in New York City and you're hesitating to decide whether you're going to walk out and a taxi comes barreling down toward you, you either flee across the street or you freeze. But that is CRF being released, not in the hypothalamus in this case, but in the amygdala, an area in the temporal lobe that I'll tell you about in a minute. And this brings me to the term pain. So there are pain pathways in our bodies. They're well worked out. Neurology has done an extraordinary job of understanding the pain pathways in our body and brain. And it involves nociceptors in the periphery, peripheral nerves, the spinal cord, and ultimately the brain. but there are two circuits. Without going into great detail, there are two circuits. And one goes through your somatosensory cortex to give you that physical feeling of pain, but another circuit, a separate one identified by a bunch of French researchers in Paris, led by Jean-Marie Besson about 10 or 15 years ago, goes directly to your amygdala, to that brain stress area. And so pain can be colored by emotional states, memories, expectations, presumably all linked up to this stress axis that I've been arguing about. So physical pain, I always tell people, can cause emotional pain, and emotional pain can cause physical pain, and they converge in this brain stress area called the amygdala. So what does this have to do with addiction? And why am I giving you this basic information? Well, we can define addiction in many ways, and the first part of this definition up here is in fact what most people would say, a chronically relapsing compulsive disorder, loss of control over drug intake. But I add on, there's a Koob add-on to this definition, and that is the manifestation of a negative emotional state when the drug is removed. And I mean dysphoria, I mean emotional pain. And I'm going to further argue, and I call this the dark side of addiction. And so the rest of my lecture is going to focus on how does this occur, why is it important, what's the interaction between reward and stress to produce this dark side of addiction. And the bottom line is this, I could stop the lecture here. I'm going to argue that addiction is an incentive salience disorder. I'm going to argue that it's a reward deficit and stress surfeit disorder. That means too much stress. And what's particularly important for you all and your stage in life, addiction is an executive function disorder. So I'm going to touch on all of those very quickly in a very, I hope, palatable way for you, and then I'm going to stop. So the way I came to this realization about addiction was through actually teaching. I taught 1,000 undergraduates at the University of California, San Diego about drugs and addiction-like behavior for 30 years, all right? I couldn't go to any restaurant in San Diego County without one of my students serving me, and I always hoped it wasn't one that I gave an F to. Anyway, what we came up with through a course that I designed called Impulse Control Disorders was that addiction and the addiction-like phenotype has 3 stages. The binge intoxication stage, which is kind of self-explanatory, the withdrawal negative affect stage, which I think is relatively self-explanatory now that I've told you that that's, the major component of that is feeling terrible, and the preoccupation anticipation stage, which comes from social psychology, which is really the craving stage. Now, but how did we get there? Well, what I realized by going through the old psychiatric literature was that we labeled disorders in the old days 2 ways, impulse control disorders and compulsive disorders. The blue over here is the impulse control disorder and, circuit, and the archetypal syndrome is kleptomania. So Lobster Man is one of the case histories they use. This chap goes into the supermarket. He starts sweating. He's got his overcoat on. This tension has been building up. He can't control himself. He goes into the supermarket. He's cruising up and down the aisles looking for where the lobster is sold. He finds where the lobster cans are. He stuffs them into his pockets. He exits the store, huge amount of relief. He feels so good. He's copped those lobster cans again. Then he drives home and he drops them in the closet because he doesn't eat seafood. And that's, kleptomania. And you say, well, that doesn't really happen. Well, it does happen. There have been anecdotes of famous Hollywood stars shoplifting, and they have plenty of money to pay for the goods. So it's for the thrill, it's the risk-taking, the impulsive type behavior. A compulsive disorder, in contrast, is like obsessive compulsive disorder. And if you've never seen the movie As Good as It Gets with Jack Nicholson, it's a perfect description of obsessive compulsive disorder. And by the way, no one does psychopathology better than Jack Nicholson. So if you don't believe me, go watch The Shining. In any event, in OCD, there's delusions or obsessions about cleanliness. There's a great deal of anxiety formed. repetitive behaviors developed to relieve the anxiety. There's no fun at all in obsessive compulsive disorder. So you take these two together, and you can see where I'm going. You have a combination of an impulse control disorder and a compulsive disorder. That leads me to one more psychological construct to pull this all together. I told you what positive reinforcement and reward was. One of the concepts that you did get in Psychology 101, but probably went in one ear and out the other, is the construct and concept of negative reinforcement. And negative reinforcement is, in fact, where you increase the probability of response by removing an aversive stimulus. So if you're working at some job here in town, I'm not going to use your professors as an example here, I'll use your boss in some job, and all they do is ever yell at you, you're probably going to avoid them like the plague. And that's negative reinforcement. The increased probability that you're never going to interact with your boss is because all they ever do is yell at you and put you down, all right? Negative reinforcement becomes a critical part of the addiction process. And so this is a real human brain, and this is the real circuitry, and I'm going to argue that there is specific parts of our brain that are involved in incentive salience, releasing of dopamine and opioid peptides. I'm going to argue that there's other parts of the brain, the red parts here, that are important for stress and negative reinforcement. And I'm going to argue that the cortex, particularly the frontal cortex, is a key part of executive function and decision making that is also part of this circuitry. But instead of a fried egg, this is your brain on drugs, all right, okay? Now, let me break it down a little bit, and in a more simplified form. So what I'm arguing is that the binge stage is basal ganglia, the blue there, and I told you that reptiles have that. And the red stage is the central nucleus of the amygdala and the amygdala connections. And reptiles have that too. But the craving stage is cortical, frontal cortical, and reptiles don't have that. So I just said that. So basal ganglia is the binge stage. The withdrawal stage is amygdala, extended amygdala we call it, and the craving stage is prefrontal cortex. Now let me break it down a little further in the simplified form. If we were to take the binge stage, we're going to argue that this is where you experience reward, the high, the intoxication of drugs, and we know that dopamine is released and endorphins are released. All major drugs of abuse, including alcohol, release dopamine. Some release dopamine massively. I don't know whether you realize it, but if you did a pipe of methamphetamine, and I do not recommend it, you would increase the dopamine release in your basal ganglia by 300, 400%. Okay? You know, that pizza probably caused a release of about 10 to 15%, just to put it in perspective. Other drugs, like alcohol and opiates, interact with the endorphins. And alcohol is particularly interesting because it activates all parts of the reward system. Not as dramatically as methamphetamine for the dopamine side, but more dramatically than methamphetamine for the endorphins. And in fact, one of my colleagues, and this is the most complicated circuit diagram I'm showing you, but one of my colleagues, Eric Nestler, drew this many years ago, and I just put it up to show you that the focus for the rewarding effects of drugs of abuse is on the nucleus accumbens. And we have the opioid peptides or endorphins, we have the dopamine, and you can fit in whatever your favorite drug is on here, and you can see how it activates ultimately these special neurons in the basal ganglia. So we've learned an enormous amount about how the brain codes reward, believe it or not, from the study of addiction. But that's not really addiction, is it? It's just feeling good when you take a drug. And I put this in to show you that this has been demonstrated in humans. So without going into great detail, a decrease in binding here means that there's an endogenous release of endorphins. And this is a person, a human being, whose brain was imaged while they were drinking 2 glasses of wine. And you can see there's a pretty dramatic release of endorphins in the nucleus accumbens, and there's an actual real picture of it. This is a real study. And how does that work into incentive salience? Well, we know that the basal ganglia have circuits in them that then encode into habits seeking that pizza for every common fund. So that by the time you finish at Franklin and Marshall, you're going to associate pizza with great lectures at the common fund, all right? Maybe. And I just wanted to take 1 minute to remind you about something about alcohol. It is something I discussed with the president's office this morning. It's a big problem on college campuses, and one of the things that I think is often forgotten is that alcohol can be lethal. It can kill you. And the blood alcohol level that kills you is somewhere around 400 milligram%. And 1 glass of wine puts you at somewhere around 20 milligram%. And 80 milligram% is that reason that you get a DUI if you're driving while intoxicated, and that's where you start to see impairments in function. But I just want to, I don't like to preach too much, but I want to remind you that if you drink 15 drinks in a 2-hour period, you are at what we call the lethal dose 50% range. I actually had someone from Slate magazine call me up and asked me what I thought of someone who drank a whole bottle of scotch in 13 seconds. My answer was idiocy. And then my next answer was Russian roulette. So I want to remind you that excessive drinking leads to all kinds of problems. You've probably heard this before, but I think it's worth reminding everyone in a college setting that and you can remind your parents for that matter, there is a dose effect curve with alcohol. Many, many people in the United States, 70% of the people in the United States drink, 90% of that 70% enjoy alcohol and the pleasurable effects from it. But we need to keep in mind that there is a dose effect function and it's a U-shaped curve, and that means that past a certain point, it only goes downhill, so to speak. Now, I got a couple minutes before I'm going to stop. I want to stop at 1:15. I want to talk about the dark side for a second. This is my all-time favorite painting. It hangs in the Hermitage Museum in St. Petersburg, but I've seen it on tour twice. It's a Picasso, and it illustrates somebody drinking a very potent drink at the turn of the 19th to the 20th century, in the late 1800s. Which was called absinthe, which contains not only alcohol, but a psychostimulant. And the dark side of addiction really focuses on this part of the brain deep in your temporal lobe. This is off to the side, sort of in toward from your ears, that we call the amygdala. And this is a part of the brain that we now know encodes dysphoria, fear, malaise, irritability, and pain. And we now know that there are neurotransmitters that encode these changes, one of which is CRF, which I told you about already, but there's a new one on the block that I'm personally really intrigued with, and my current laboratory is actually still working on, called dynorphin. And I want to remind you that in addiction, this dysphoric stage is an acute response to withdrawal, as illustrated here in a classic study done by Mark Schuckett and Sandy Brown on alcoholics. But even though mood returns toward normal in alcoholics, 4 weeks out, they're still not really tiptoeing through the tulips. You have to take my word for it. Tiptoeing through the tulips is where your Hamilton score is down here, not up at 10. And there's been classic work done by the director of the National Institute on Drug Abuse, Nora Volkow, my colleague at NIH, showing that there is a dramatic decrease in dopamine function, one of your reward transmitters, when you're in this withdrawal negative affect stage. And again, this is an imaging study. It's difficult to explain, but this is D2 receptors. They're given methylphenidate as a challenge, and the control subjects show this decrease in binding, which indicates that there's dopamine being released. The alcoholic subjects not only have a decrease in dopamine constitutively, but when they're challenged with methylphenidate, there's no change here in this direction. This is a dramatic example that she's repeatedly shown now, and many other investigators, that when you are addicted to drugs of abuse, your dopamine system's not working so well anymore. And so I told you that these guys are activated when you get high, but when you stop taking the drug, your reward system is compromised. I'm going to repeat that. Your reward system is compromised. It's not functioning normally. And your stress system is recruited. So it's a double whammy. Your brain's emotional systems have been dramatically dysregulated. You lose your reward system, you gain your stress system, and that's why you feel so lousy. And believe it or not, that's also the basis for a hangover, which is young people, most of you don't have much of, but believe me, you will. CRF is one of the players. I told you about that. It's activated in the extended amygdala. But one of the more insidious parts, and again, this is complicated, maybe only the neurobiology people will understand this, but when you overactivate the dopamine system and the opioid peptide system, you cause a molecular change that increases the amount of dynorphin, which I said was one of the stress transmitters, that feeds back and shuts off the dopamine system. So it's worse than what I told you. Not only do you recruit the stress system, but the stress system is shutting off your reward system. Think about that. Why would it be that drugs of abuse who make you high then cause this activation of the stress system to shut off your reward system? It's almost as if there's a built-in brake to your brain's emotional systems. And so this is an illustration of what happens. You lose the reward system, you gain the stress system. Finally, just a mention of the craving stage. One of the things that's important to impart upon you, and your professors know this, but your frontal cortex is not fully developed until age 25. This is the part of your brain you use for executive function. It encodes desire for the drug, sleep disturbances, emotional instability in protracted abstinence. And the transmitter that we know is disrupted and dysregulated is one called glutamate, which is the major excitatory transmitter in the brain. One of the things we know is that excessive use of drugs, any drug, probably includes marijuana. The data are only just coming in, but excessive use of any drug during adolescence disrupts the development of your frontal cortex, slows it down, may permanently reduce its function. And this is a real concern because we're beginning to think that this might be the key for why your reptile brain that I was talking about gets so engaged, why your reward transmission system, the basal ganglia, gets overactivated, and then your stress system kicks in. And in fact, there's very strong data out there showing that the more you drink, the less cognitive functioning you have. And this is the drinks per drinking day. The black are individuals with a substance use disorder, alcohol use disorder, and you can see there's a pretty good correlation between impairment in cognitive functioning and how much you drink each day. And just a reminder that it's not facilitating your ability to perform in an educational institution. So this is kind of a brief, maybe intense overview, but I'm arguing with you that drugs of abuse have three major functions, and they're regulated by three major areas. So there's a reward, incentive salience effect, habit formation that leads to compulsive behavior during the binge intoxication stage. That's followed by activation of the brain stress system's loss of the reward system, and that's the withdrawal negative affect stage, and at the same time, your frontal cortex isn't working so hot. So these are my conclusions. Why do we care about reward and stress? Well, addiction is a disorder of reward and stress and wreaks havoc on the fabric of our lives, costs the US about $500 billion a year. It's a major burden to our healthcare system. The brain reward system, brain reward circuits are key parts of our reptile brain and guide us to incentives of value for well-being, and we feel good in the process. The brain stress systems or brain stress circuits are key parts of our reptile brain and guide us to avoid dangerous, painful, and aversive situations, make us feel bad in the process. The brain cognition systems, the brain executive function circuits control our reptile brain to help us make choices, make decisions, plan for the future, and prevent us from being impulsive and compulsive. So what did you learn here today, hopefully? Well, reward and stress are the two faces of Janus. Stress can be rewarding in low doses, but can drive compulsive behavior in high doses, or negative reinforcement, as I argued, taking the drug to avoid that terrible feeling. There's no free ride in the brain. Excessive activation of the reward systems can lead to stress and anti-reward. These systems are in homeostatic balance. The frontal cortex does not fully develop until age 25. Drugs of abuse impair frontal cortex functioning and delay development of the frontal cortex. Controlling impulsivity and compulsivity is a key role for the frontal cortex and needs to be cultivated. To quote Voltaire, you need to cultivate your garden, but the garden that I would cultivate is your frontal cortex, strengthening the ability to make decisions, delay reinforcement, and think through problems and choices. I made an argument in the back of a science paper up in the right-hand corner of the notes one day, many years ago, where I argued for a concept called hedonic Calvinism, which is obviously an oxymoron. And I argued that it was a framework for happiness. Engagement of your brain reward system, I argued, was a limited resource. Use it wisely and strengthen these systems, just like you would strengthen a muscle. So some narcissistic self-promotion. We have a book that's now, we hope is a textbook, at least be utilizable by graduate students, maybe undergraduates, called Drugs, Addiction, and the Brain, where I explain many of these concepts that I just discussed with you today. And the Alcohol Institute has a lot of publications. So if you have a loved one that has an alcohol use disorder or a problem, I highly recommend a number of these publications. You can find them on our website. One in particular that's quite popular is Rethinking Drinking. There's a calculator in there. You can calculate how much alcohol is in any given drink or any given mixed drink. You can find out how many calories are in your drink. You can find out what the dose effect curve looks like. And there's a lot of other interesting factoids in there. The calculator you can actually interact with and get your information. So thank you very much. So I'll be happy to answer questions. If they're personal, I won't answer them. But if they're anything about drugs, I know more about drugs than most people. So if anybody's got any questions, I'm happy to answer them.
Yes, hello. Is this on, or? My question is, many rehab facilities boast a 25% success rate over a year's time, which I assume is hard to measure, but that seems to be a common number. My question to you, sir, is what do you feel is the reason that that number is as low as it is? Who or what is at fault for such a low number of success rate? Thank you.
Well, that's a really good question, and the simple answer is we don't really know. But given that I am me, I will give you what I think is playing a role here. And one of the major issues is that most rehab setups do not follow the patients after they're released from a 30-day treatment program. And if they do follow them, it's fairly cursory. One of the things we're going to be looking at in our new strategic plan for the Institute is indeed recovery. We don't even know the numbers for a fact. We don't know how much follow-up is done, and we don't know the numbers for what works in follow-up and what doesn't. But I think it's important that you all realize that there are 3 medications on the market for treatment of alcoholism. They have modest effect sizes. They're not a whole lot different than an antidepressant in effect size. And we have some very good behavioral treatments. Cognitive behavioral therapy, for one, is excellent. Motivational interviewing works quite well. Even brief interventions with primary care physicians seems to work, particularly in youth. And family therapies are often very useful, woven into the context of these other behavioral therapies. So my dream team kind of treatment facility would be one that does the detox and the 30-day treatment program, should it be needed, but there would also be then a follow-up where the individual joins Alcoholics Anonymous and goes. Alcoholics Anonymous doesn't work if you don't go. Maybe also has counseling sessions, maybe also has some individual psychotherapy or cognitive behavioral therapy sessions. I think we really need to look at that period, and it may need to extend past 1 year. It may need to extend to 2 years. Generally, you know, I only go by rule of thumb because there's no other data, but if you talk to really heavy cigarette smokers who have kicked the habit, so to speak, somewhere around 12 to 18 months, they have this switch that turns where they don't want to be around cigarette smokers anymore. They don't want to have anything to do with smoking. And those individuals generally stay abstinent. So those are my thoughts on that one. Other questions? Yeah.
Hello, Dr. Koob. So you mentioned that our frontal cortex does not develop fully until the age of 25. So I was just wondering, are there any exercises or stuff like that you would recommend for young people to cultivate their frontal cortex? Thank you.
Yeah, well, that's a good question, and I don't completely fully know the answer to it, but I would say that going to class, doing your homework, I could tell who the faculty are applauding at least. Reading, engaging in exercises that challenge decision-making, delaying reinforcement. These are all things that are part of it. Most of you are probably familiar with the marshmallow experiments, right? You know, the famous work showing that 4-year-olds who could delay reinforcement at that age then go on and have better SAT scores, do better in college, have less divorces later in life, and the ones that couldn't delay reinforcement generally are in the other direction. And believe it or not, that work was followed up with imaging 40 years later, and the ones who could delay reinforcement were the ones whose frontal cortex was working more functionally and more normal, quote unquote. So I'm always block on this gentleman's name that did all this pioneering work, but one of the things they're working on are paradigms that will help strengthen the frontal cortex during development. And I think this is an important area. Some of you may be working in this or planning on working on it for your careers. I think it's a great area of work, and it's certainly something at the Alcohol Institute we're interested in because of the cognitive impairments that we know are associated with fetal alcohol syndrome and fetal alcohol spectrum disorder. That's a really good question. I'm not the world's expert on stimulating your frontal cortex, but in gross, those are some of the issues.
Hi, I'm Courtney. I'm a junior, an American Studies major. So my question has to do with binge drinking in a college setting. So someone who is a college student and, engaging in binge drinking, what are the chances that people like that just stop their habits once they graduate? Is there a lot of carryover in their adult life, that sort of thing?
Yeah, so that's an important issue, and the reason we're so focused on underage drinking and binge drinking is because so I'm going to turn your question around and say, what's the problem with binge drinking for later in life? Well, one is the development of the frontal cortex and the fact that you may end up having a more difficult time strengthening your frontal cortex and learning not to be impulsive or compulsive, controlling your stress systems later in life. But a more concrete answer is that we know that individuals who start binge drinking really young, like 11, 12 years old, have a fourfold higher probability of an alcohol use disorder later in life. And by the way, that holds for all drugs of abuse. Now, it's a linear decline. By the time you get to 21, the likelihood is probably twofold. But you know, there's really, as far as I can see, no benefit to binge drinking, and only negative. You lose control of your function. The tolerance develops within a bout of drinking. Everybody forgets about that, but the first drink can produce those beneficial effects. I call it social lubricant effects. You know, I admit, you know, the Research Society on Alcoholism starts the meeting with a cocktail party, okay? Why? So the researchers will go down and love each other, all right, and talk to each other, and interact. Probably they assault each other from time to time because somebody stole their idea, but I will leave that behind, all right? But the fact is that alcohol has lots of beneficial effects, but getting so intoxicated you can't move is not one of them. And in fact, it's the other direction. And it can be life-threatening. People who binge drink regularly don't perform as well academically in general. Yeah, I always hear about the guy who was stoned all through college and still got into Harvard, you know, graduate school with a 3.9, but they're the exception to the rule, trust me. So I think that the best thing is to learn when you're 21 to drink in moderation and enjoy alcohol for what it was intended for. We're not prohibitionists or neo-prohibitionists at the Alcohol Institute. In fact, a week doesn't go by that we don't get a phone call from the beverage industry on one complaint or another about what we're doing or what they think is being done to them. But we do interact with them, and they do support our work in binge drinking and the deleterious effect of alcohol. So, I do want you to understand that I really believe that alcohol can be used in a responsible way, but it is something that needs to be learned in a proper way. Any other questions?
Yeah, hi, I'm Sean Flaherty, economics professor here at F&M in Don of Wise College House. As a cranky old man, I'm tempted to ask if the less than complete development of the frontal cortex has anything to do with the behavior of young people when they get up and leave lectures and, at the question and answer period. But that's not my real question.
No, the pizza wore off.
We know the notion that there are happy drunks and mean drunks. I'm wondering if is there a biopharmacological explanation for those differential responses to alcohol intake?
That's a really, really good question, and it's something that we're looking at. I talk about it. I'm a lumper, as my colleagues have accused me of being, which is I tend to look at for common elements in addiction, common elements in alcoholism. But these are very diverse disorders. The phenotype is very different from one person to another. And every one of you probably has a family member somewhere in the family history tree who is what you said, a mean drunk or the jolly drunk. But often it can be the same person too, I might add. It just depends on how much alcohol they had and the context. And of course, family gatherings can turn a happy drunk into a mean drunk because somebody gets insulted because of the disinhibition that occurs. But we are looking at these individual differences, and we do believe that they may be the key for treatment so that we can tailor treatment in the future to the mean drunk. Might be different. Maybe it's their CRF system is hyperactivated, whereas the jolly drunk maybe just have an overactive basal ganglion, and that may require, say, naltrexone as opposed to a camprosate. So this is actually being looked at. In some sense, Francis Collins, the director of NIH, I believe it's today, they're loosing the precision medicine plans for the institute, for the institutes, there are 26 institutes. And this falls under that rubric of precision medicine. We'll be doing genetic studies, we'll be doing epigenetic studies. Are there history of the organism? In other words, if you were in an abusive family, which we know, and that could be any kind of abuse, from sexual abuse to physical abuse to living in a war zone, that those can contribute to an alcohol use disorder later. Is there a specific gene that gets activated by those stressors that then carries on to make that person a mean drunk? That's part of our strategic plan. It's a really good question, but right now we don't have an answer. But we have the tools, I believe, with modern medicine to get the answer, and so that's what we're hoping to do.
I was curious if there's a genetic component to alcohol abuse and alcoholism, and if so, how does that relate to the biology that you described earlier?
Again, that's a perfect follow-up question, and the answer is yes, there is a genetic component. Some of you already know that if both your parents were alcoholics, you have like a 4 to 5-fold higher likelihood of becoming an alcoholic yourself. There's also a social component to that. Sandy Brown always used to tell me that daughters of alcoholics tend to marry alcoholics. What is the basis for that genetic change? Well, we know there's a phenotype associated with it, and that tends to be the people who have a low sensitivity to alcohol. It's just the opposite of what you would think. So the people you knew when you grew up with who could drink everybody else under the table, those are the people who have a higher likelihood, if they have a family, often have a family history positive for alcoholism. And the guy that did all that work is Mark Schuckert. He's still working at it at the University of California, San Diego. He's famous for this work. What are the genes involved? Well, that's a really good question. We know that there's a couple that may be involved. One is a peptide that works in concert with CRF. It's called neuropeptide Y. That's actually a mellowing out peptide. A couple of genes in the GABA system, which is the major inhibitory transmitter in the brain, are candidates for these differences. And more recently, there is a gene that's on the promoter for growth factors in the brain that people are hypothesizing may be involved. But again, this is an area of intense research right now at the Institute, and a really good question.
I'm Virginia Maximowitz. I'm a professor of sculpture here, and I also happen to be chair of the Common Hour Committee. And I have a question about the reward center in the brain. We've all heard of runner's high, and I believe there was some research done at the University of Pennsylvania where nuns volunteered to have their brains wired up as they prayed. And they were able to image certain parts of the brain being stimulated. Do you know of other research that's been done where non-drug rewards could be actually measured in the brain?
Yeah, that's a really good question. The converse has been demonstrated in a few disorders. So individuals with internet addiction, compulsive gambling, compulsive food eating have shown those decreased D2 receptors that you see in addiction. But the converse of that, which would be, can you strengthen your reward system, and can it be demonstrated with imaging? I'm not up to date on, so I have to say that there may be some studies. I'm not familiar with I vaguely remember something about the nun study. My argument would be that like any other muscle, in our body, the brain muscles can be strengthened. And I think that appropriate use of rewards and it will actually allow you to enjoy life a little better and enjoy the natural rewards a little better. You know, one of the therapies that's used and is more and more demonstration of effectiveness in a number of mental disorders is yoga, for example, okay? So, and one of my colleagues at the University of California, San Diego, that I used to play racquetball with, is a yoga and has published on this and utilizing yoga for therapeutic purposes. But I've talked to him on many occasions, and he'd love to do an imaging study, but getting the funding for that has not been forthcoming. So, you know, and when it comes to priorities, it just hasn't reached that level. Tell your congressman, no, I didn't say that. I'm not supposed to solicit funding. Other questions? Yeah.
Hi, I'm Sarah Black. I'm a historian of drugs. And I was just wondering if you could talk a little bit more about this sort of pain and pleasure concept, the sort of Janus-based nature of addiction that you were talking about, specifically in terms of withdrawal from drugs that are sort of more pain relieving and sort of addicts' sensitivity to pain than in the withdrawal stage. Thanks.
Yeah, well, that's another good question. And somewhat neglected, although both Nora Volkoff and myself are quite keen on studying pain responses and how pain responses can be part of the relapse phenomenon. There is a huge problem, as you know, with prescription opioid abuse in this country. Nora is called down to Congress on a regular basis to testify about this issue. They're trying a number of things. But I think one of the things that I would say is that opioids can cause pain, and that may fly in the face of everything you know about pharmacology. But what I'm arguing is that opiates, or opioids, I call them opioids, that's the more generic term these days, can cause pain. Now, what do I mean by that? Well, opioid drugs have relieved more suffering than any drug in the history of mankind, and they probably saved more lives. They saved lives because they block dysentery and gastrointestinal transit. And so in many areas where there's an epidemic of dysentery, which will kill young children, the opioids are life-saving. And of course, they relieve pain, and all of you have probably, if you broke something, had received an opioid. And they're marvelous for that. But where we get into trouble is when we are given chronically. And so if you give an opioid chronically and the dose goes beyond what's needed to control the pain, there is an opposite reaction that's called hyperalgesia. My colleague Guy Simonet at Bordeaux, the University of Bordeaux, studied this for years, and everyone ignored him. He was admittedly, he was using animal models, and he was seeing when he would give an injection of fentanyl that, you know, if you look 2 or 3 hours later, he was getting a hypersensitivity to pain, not a hyposensitivity to pain, but a hypersensitivity to pain. Well, Guy prevailed. It wasn't until toward the end of his career, but now anesthesiologists are beginning to recognize that hyperalgesia with chronic opioids is the phenomenon and a marker for a problem. So the simple solution to this is one that I think is starting to be engaged, or at least one of my suggestions for a simple solution, which is something I've argued for a long time. When you go on opiates and you take them chronically for pain, taper them off. Taper. In other words, cut the pill in half and then in quarters. You are going to feel some withdrawal. You're going to be uncomfortable. But it doesn't necessarily have to be really uncomfortable or drive addiction. And the other good reason for tapering is, how do you know that the pain that you are treating is still due to the injury or is due to the fact that you're still taking the opiates? Think about that. So, you know, there are formulations that can be used that can prevent some of the overdose, and I'm sure that's what Dr. Volkoff and the government's working on. But I think there's some physiological things we need to learn. And by the way, alcohol is a tremendous pain reliever. Just ask grandma. And so the same issues kick in.